Thank you very much for having me here today. So, as Amitabh was saying, thanks for inviting me. One of the things that I'm trying to do here now is trying to bring this around full circle for us. We've been talking a lot about patents-related endpoints, and as Danielle was just showing you, one of the things that we can see there is, well, you get a lot of patents, for sure. We do know that for sure. You get a lot of patents when you fund, when NIH funds research. But how much does that actually translate into new drugs, and how much are those new drugs actually effective for the end users, for the patients, and improving their quality of life? So there are many studies out there that are providing that sort of descriptive evidence. And as Danielle pointed out, one of our big concerns as economists coming towards this is that we want to know that there's some causal linkage, and we want to know that there's not crowding out. So first, thinking about what is the role of NIH research? Again, Danielle and Bob both really set the stage for this a lot. The information that NIH grants are providing whenever a paper gets published, when research results are transmitted at a conference, that information is obviously very important. So is the training of the biomedical workforce. That's another cost-reducing subsidy that NIH is providing to the pharmaceutical industry. The big problem that we see is then, well, what's the counterfactual? If NIH weren't doing this, does that mean that the drugs wouldn't be developed? Does that mean that people wouldn't pursue PhDs? It turns out actually quite a large number of biomedical sciences PhDs aren't funded by NIH. There are other people that are funding these graduate students. And so we need to know to what extent that crowding out is occurring, where public money might be getting in the way and actually pushing out private sector investment. And so I can say, consider a kind of a mental experiment. Suppose that NIH's budget for research and development, for extramural research and development, expanded 10 times, 100 times. We could, in principle, potentially, to the extent that NIH research is a substitute for what the private sector does, completely do all of the biomedical research that needs to be done for the pharmaceutical industry. And then, through the descriptive exercises, we could trace back and say, yep, every single drug that is going to market was developed with NIH-funded research money. Does that mean that it wouldn't have happened had NIH not stepped into the gap? No, and so this is what we need to tear apart, and that's why this emphasis, again, as you heard from Danielle, on more sort of causal approaches to trying to figure out, okay, what is NIH doing that wouldn't otherwise be done? So I'm going to be talking also about an older paper that was published back in 2012, but I'm looking at a very specific policy lever that Congress might like to have NIH work on. For example, let's suppose that we say, oh, well, there aren't enough good therapies for brain cancer. Let's increase brain cancer funding by $100 million. Do we think that that's going to increase the number of drugs to treat brain cancer down the road? And as you've been hearing, it turns out there's actually a fair amount of evidence for spillover effects, for serendipitous occurrences. So this is what my paper was exploring. The research questions I was asking specifically looked at NIH extramural research funding. Most of this is being conducted by U.S. universities, medical schools, and so forth. The outcomes I'm looking at are not the patents, but the actual R&D activity. To what extent do phase 1 trials, so those first clinical trials in humans, start occurring? To what extent do we see an increase in the number of drugs then that make it all the way to phase 3? You've already seen this in Dr. Austin's talk, the sort of pipeline sort of idea. The outcome variable, again, that I'll be looking at is clinical trials. The data are coming from pharma projects. This was before clinicaltrials.gov gave us all of the information that we have today. So a lot of the things also that you'll be seeing, that I'll be pointing out, are things that would make it a lot easier, because the data are now available that weren't available before, to do this sort of study, if somebody wanted to update it. The variables I extracted, I was looking at phase 1 and phase 3 trials. I'm specifically looking at individual therapeutic classes, what trials are getting started in any given year. Oftentimes, you have a molecule that's actually being explored for multiple different indications. Those might be in different phases at different points in time. I'm capturing all of that. Originally, back before NIH Reporter, there was CRISP, the Computer Retrieval of Information on Scientific Projects. It allowed for a Boolean search using and, or, not, things like this with keywords to try to find out how many grants actually are related to any particular disease. But there are downsides to that sort of keyword search. NIH Reporter still allows, you probably can't see down in the box, the text search box down at the bottom, you can do an advanced Boolean search, but it basically comes down to the same thing, this idea of looking for specific keywords. The keyword approach is a problem in biomedical sciences. This is sort of a known problem in bioinformatics. Because oftentimes in the biomedical domain, 6 or 7 different words might be used to describe the exact same phenomenon among experts in that field. More than that, though, a lot of times if you're looking at a particular NIH grant abstract, you might find that the particular disease or condition isn't what's named. Maybe it's the organism, some sort of cellular anatomy, something about the molecule, where anyone who's expert in that area would say, well, obviously, this is a breast cancer related grant. But if it doesn't actually say that anywhere in the grant, then it's not going to get picked up through any sort of keyword searches that are looking to categorize that as breast cancer related research. The specific example that I'm showing you today is actually from malaria. So malaria, the protozoan associated with malaria is Plasmodium falciparum is one of them. And yet, if you do a Boolean search for malaria, and then alternatively do a Boolean search looking for Plasmodium falciparum within NIH Reporter or previously within CRISP, you would come up with sort of this, like the Venn diagram. There'd be a lot of overlap, but those were not completely overlapping sets of returned results. So this is what my approach was meant to sort of deal with. As Danielle was talking about, we're able to leverage the Medline subject headings index to try to deal with this problem. For each mesh term, for example, malaria, you have a whole bunch of different entry terms which can be viewed as synonyms. Among them, you can see, is Plasmodium infections. So one of the things that I was doing was calling all of those synonyms for each of the words so that if somebody mentioned in their grant abstract not malaria but the Plasmodium infections, I would pick it up as a malaria-related related grant. And then, of course, C also anti-malarials. Has malaria as a root, but it's good to have it explicit. Also, alternative names, marsh fever, or if we go down further and start looking at the hyponyms, so the special subcases. Another example might be within arthritis. There's chondrocalcinosis, as well as osteoarthritis. If I'm just looking for the root arthritis, I'll catch osteoarthritis, but not chondrocalcinosis. So being able to break that down further and look for things like blackwater fever. That's all getting captured in the algorithm that's being used to assign each of these to specific diseases, each grant to a specific disease. So I won't go into the details here, but I'll be happy to speak with anyone who's interested in knowing how to replicate this. But basically the idea was to scan for all of those keywords, the synonyms, the hyponyms, to try to figure out to what diseases or conditions should this grant research be assigned. Then comes the econometric estimation. There's a lot of technical mathematical detail behind this and doing it in a way that deals with possible feedback effects. For example, there's the possibility that if NIH funds more research this year, there's more drugs next year. But if there are more drugs next year, does that cause there to be more NIH research in year T+2? If there is that sort of feedback effect, that's something that we need to deal with mathematically and statistically. So I'm dealing with that. I'm dealing with the possibility, like Danielle had brought up earlier, of a push, a shock, some kind of thing that affects both federal and non-federal funding in the same year, causes them both to go up. And I'm also looking at just, well, you know, some disease areas have a bigger patient population, or they're maybe more tractable than others. There's some that are going to just have a higher average number of clinical trials happening, just on a year-to-year basis. The analysis deals with that as well. So what I'm looking at ultimately is if NIH funding increases, and I'm allowing lags, in this case, up to 22 years prior for a particular disease, do we see more drugs entering clinical trials to treat that disease? Do we see more in phase 1? Do we see more at phase 3, which are the more expensive randomized trials for efficacy? And I'm controlling for differences in market size, which is important because if we're thinking about market size, and this is looking forward market size, for example, with demographic boom, with aging happening in the population as a whole, we might see more of a sort of movement, both from NIH and from the pharmaceutical industry, to treat diseases that primarily affect older people. Looks like I need to speed up a little bit here. So what did I find? Well, I did find that if you have a sustained 10% increase in disease-specific R&D funding, you'll see a 4.5% increase in the drugs entering phase 1 trials to treat that disease after lags of up to 12 years. But this does not persist to phase 3. We don't see any increase in the likelihood of success when you have those new drugs coming into phase 1 trials. We don't see any apparent effect of NIH R&D funding on supplemental indications for the for drugs that were already approved for some other disease. So the phase 1 effect that we see there is actually consistent with a lot of other studies that look at the sort of impact on innovation of R&D expenditures. The phase 3 results, though, I want to be very careful about what that does and does not say. First of all, I was only looking at traditional, typically investigator-initiated extramural research project grants. I wasn't looking at clinical research centers, translational research, traineeships, fellowships, intramural funding. Also, very importantly, I was looking at the effects of changes in funding. If we change the current level, does that change the flow of drugs? This isn't saying anything about the total or the aggregate number of drugs and the extent to which NIH contributes to that total. And focusing on this policy lever, which I think is important, is something to communicate to policymakers, but focusing on the policy lever explicitly ignores the spillover effect that you've already heard is important. So this is a lot of detail that I probably don't have time to go into, but just to give you sort of an example of a cancer drug and the various roles that NIH played along the way in its development. The patent that was originally associated with the drug, which is not one of the covering patents that Bhavin and Danielle would have found looking in the Orange Book, but the original patent was filed back in 1989. The publication associated wasn't published until 1994. The 1989 patent doesn't cite any publications, but the 94 publication has 3 key references, all of them to NIH-funded research. You then go forward a bit, achieving total synthesis of that particular molecule. That was done by an NIH-funded postdoc. But still, we're not talking about a specific disease or condition with respect to this particular molecule. Ultimately, the company that held this patent sort of stepped away from it because cardiotoxicity looked really bad in their animal trials. They said, we're not going to continue with this. That's where NCI stepped in and started actually doing intramural research and found a way to make that safer for human use. Ultimately, it was that NIH research that caused private sector to step up and start doing the sorts of clinical trials that we then see. And ultimately today, the drug was acquired and marketed by Celgene in 2010. There is still research going on with respect to this drug by NCI in combination with other therapies looking at other endpoints. The reason that this is important is sort of thinking about what are the many different roles that NIH is taking in this. Certainly there's the fundamental science, but it's also the communication of results. Every time NIH researchers had, those NCI intramural researchers had a result, it was getting published. The publication associated with the original patent was 5 years later. So the patent was out there, but it took a while. Training the biomedical sciences workforce. It was an NIH-funded postdoc that said, hey, look, we can synthesize this. Considering things where it's high risk, small market, long window, translational sorts of research, there was so much failure that went on here. Phase 2 trials for leukemias, lung cancer, renal cell cancer, prostate cancer, colorectal cancer, no effect or very little. And yet serendipity occurs, and it turns out it's useful in T-cell lymphomas. So this is that sort of serendipity in a kind of high-profile case here is actually something that we need to consider more broadly when we're thinking about how to use NIH-funded research. And, of course, you've heard about the importance of teams. And the triple helix here is academia, it's government, it's industry, all working together, spurring those sorts of collaborations. That's another role that NIH took in this process. So if I may take just an extra minute or two just to talk about some directions for future research. Again, these are things that both Bob and Danielle also touched on some of these as well. But using some machine learning statistical text analysis to think more about NIH's role in the genesis of ideas. We have the countables. We can count our publications, we can count our patents, we can do the citation analysis, the bibliometrics. But what about the ideas? And that relates to the question of how do we weight those citations? How do we decide what's a critical citation? The citation without which this new paper never would have come about. How do we find that without having to read every single paper that gets published? We have more machine learning and statistical text analysis techniques that we can apply towards that today. Using the restricted and administrative data that are available to actually follow NIH students' careers. Right now, unless you have restricted data access to census data and can link data sets, this isn't really possible. But trying to figure out who are the graduate students that are getting funded by NIH, what do they choose to do later on? To what extent is that helping pharma? What are the needs of the pharmaceutical industry? Are the graduate programs that we have now feeding people who are going to work in drug development down the road? We know that NIH R&D funding has a really, really dramatic effect on graduate students' enrollment. I have a few papers that have looked at that, on the probability that a graduate student is going to stay in science after they graduate, but knowing what the effect is going to be in terms of their working in industry later on, that we don't. And finally, thinking about the effects of different funding mechanisms. So I was talking about specifically extramural research funding, but you could also consider prior research on the SBIR, STTR programs have had sort of mixed evidence of results. And apparently there's some data limitations as far as thinking about how that effect translates within NIH. Thinking about which of these funding mechanisms is actually going to be most effective in giving us useful innovation. We have these countables, these patents, but how many of them actually reflect something that is going to actually make people better off, make humans better off? And then considering specifically the influence, not just of NIH extramural research, but of translational science, of collaborative research, the CRADAs, the UO1s, these sorts of mechanisms. So thank you very much, and I apologize for going over time.